Welcome to Podcast Learn and Fun. Let's start today's journey of learning by diving into the fascinating world of cells. All those breakthroughs we hear about in medicine, new cancer treatments, advances in immunology, all that. A lot of that happens because of a tool called Fluorescence-Activated Cell Sorting, or, FACS for short.
FACS is a technique that lets scientists identify, analyze, and sort individual cells based on their unique characteristics, of course.
Okay, so it sounds like FACS is like a high-tech sorting machine for cells. How does it actually work, though? It must be so precise to work with something as tiny as a single cell.
It is, yeah, it is incredibly precise. It all starts by tagging the cells with fluorescent antibodies.
Tagging.
Yeah, they act like tiny glowing flags that attach to specific molecules on the cell surface. These molecules could be proteins, carbohydrates, or other markers, the markers that differentiate one cell type from another.
Oh, okay, so it's like giving each cell a unique barcode that, lights up.
Yeah, that's a great analogy. Once the cells are tagged, they're put in a fluid, and then they pass one by one through a laser beam.
Oh, laser beam.
Yeah, and the laser excites the fluorescent tags, making them emit light, but specific wavelengths of light.
So each cell gets its own little laser light show.
Right, so that's where the sorting comes in.
Okay.
As each cell passes through that laser beam, the detectors measure the intensity and color of the fluorescent light emitted. And with this data, the machine can identify and categorize each cell based on its unique combination of markers. Then the machine uses an electric charge to deflect the cells into different containers, separating them based on their characteristics.
Wow, so it's like each cell gets zapped with this tiny bit of electricity and sent to its group. That's incredible.
Yeah, and the speed is incredible too. FACS can analyze and sort thousands of cells per second.
Thousands per second.
Thousands per second, making it a high-throughput technique.
Right.
So this is crucial for researchers, who need to study large populations of cells and isolate specific subpopulations for further investigation, obviously.
It sounds like FACS is an incredibly powerful tool with a lot of moving parts. What are some of the key components of the machine that are essential for this to work?
The lasers and fluorochromes are essential. The lasers provide the excitation light, and the fluorochromes, which are attached to the antibodies, they emit light of specific colors when they're excited. Different fluorochromes emit different colors, and this allows researchers to label multiple markers on a cell at the same time.
Oh.
So, for example, you might use a green fluorochrome to label one marker, and a red one to label another marker.
Got it. So the machine can see multiple characteristics of each cell at the same time, like its size, its complexity, and the specific markers it's expressing. It's like having a multi-dimensional view of each cell.
Exactly. And to distinguish between all those colors, it uses optical filters and detectors. These filters select specific wavelengths of light, and the detectors measure the intensity of the light that passes through. This data is then used to identify the cell based on its fluorescent signature. Think of it like a prism, separating white light into its component colors.
Wow, that's so intricate. I'm starting to see the complexity of this technology and how it lets scientists analyze cells in such detail. What about the fluid? The fluid that carries the cells. It must be important to make sure the cells flow through the system smoothly.
You're right. The fluidic system is critical. It uses precise pressure control to ensure that the cells travel in a single file line through the laser beam. This is essential for accurate analysis because the detectors need to measure the light from each cell individually.
So we've got the lasers, the fluorochromes, the detectors, the fluidic system, all working together to analyze and sort cells based on their characteristics. But how does the machine physically separate the cells once they've been identified?
That happens in the electrostatic deflection system. Once the cell's characteristics are determined, the machine applies an electric charge to the cell.
Okay.
And this charge determines the path the cell will take as it exits the laser beam, directing it into the appropriate container.
So it's like a tiny conveyor belt, sorting packages based on their labels, except this is happening at a cellular scale.
Exactly.
This whole process is amazing. It's amazing that we can manipulate and sort cells with such precision. What are some of the ways that this technology is being used in research and medicine today?
Oh, the applications are so diverse. It spans so many fields. Immunology, cancer research, stem cell biology, drug development, the whole thing. It's It's really changed the way we study and understand cells.
It really is mind-boggling to think of the possibility. We've all heard of personalized medicine, right? I imagine FACS plays a role in that.
Absolutely. Yeah, one of the most impactful applications of FACS is immunophenotyping.
Immunophenotyping.
Yeah. It involves identifying and characterizing different types of immune cells. So this is really crucial for understanding how our immune systems work, right? And for diagnosing diseases, immune-related diseases, and developing treatments, targeted therapies.
So, how exactly is FACS being used to understand and treat diseases?
Well, take cancer research, for instance.
Okay.
FACS is essential for identifying and isolating cancer cells from complex tissue samples, right?
Right.
This allows scientists to study their properties and to test different drugs. It's also being used to develop those personalized cancer therapies, by identifying specific markers on tumor cells that can be targeted with, drugs or immunotherapies.
That's incredible. And it sounds like FACS is giving researchers a way more precise way to target cancer cells.
Yeah.
Which could mean better treatments with fewer side effects.
Yeah, exactly. And it's not just cancer. FACS is also a key player in stem cell research.
Wow.
It's used to isolate stem cells from, various sources. Bone marrow, umbilical cord blood, things like that. This is so important for developing stem cell therapies, for spinal cord injuries, heart disease, the whole thing.
Stem cell research has so much promise. It's fascinating to hear how FACS is contributing to that. It seems like this technology is leading to all these breakthroughs in medicine. But, are there any limitations to what FACS can do?
Yeah, it is a really powerful tool, but there are some limitations. One challenge is that it requires cells to be removed from their natural environment and suspended in fluid for analysis, you see. And this can change their behavior a bit, making it harder to study them in a truly physiological context.
So I see, even though we're getting all this detail about individual cells, we're still kind of limited by the fact that, we're taking them out of their natural habitat. I guess that could affect the results, right?
Exactly. Another challenge is the cost and complexity of the equipment. It takes specialized training to operate and maintain it, which can limit access for some researchers. However, technology is always advancing, making FACS more user-friendly and accessible.
That's good to hear. It makes sense that these powerful tools become more available as the technology gets better. It seems like we hear about a new breakthrough every day. What are some of the recent advances that have really changed FACS technology?
One exciting area is spectral flow cytometry.
Spectral flow cytometry.
Right. This technique uses multiple lasers and detectors to capture a wider spectrum of that fluorescent light, so researchers can analyze more markers on a single cell.
So instead of just seeing red, green, and blue, we're seeing the whole rainbow of fluorescence.
Yeah. You could say that this increased resolution gives us a better picture of, cellular complexity and lets us identify those rare cell populations. That might have been missed, with traditional FACS.
Impressive. I can only imagine what we'll be able to learn about cells as this technology keeps improving. I'm also curious about the analysis of the data. We're talking about thousands of cells per second. It must be a huge amount of information to process.
Yeah, you're absolutely right. Analyzing that FACS data can be quite challenging. But new computational tools and software are being developed to help researchers visualize and interpret this data.
So it's not just about the hardware, but also about developing the software to make sense of all that information.
Precisely. And as we get better at analyzing this data, we'll be able to unlock more and more insights into how cells work.
So exciting to think about. So what's next for FACS? What are some of the big questions that researchers are still working on using this technology,
Well, one area of research is developing new fluorescent probes that can target molecules inside cells, not just on the surface.
So we're going even further, beyond the membrane, to see what's happening inside.
Exactly. This would allow us to study processes in much greater detail and could lead to new drugs and therapies that target, specific molecules or pathways within cells.
It seems like the possibilities are endless. But, are there any ethical considerations with this level of precision and control over cells?
That's a good question. As with any powerful technology, it's crucial to use FACS responsibly, ethically. While FACS itself is just a tool for analyzing and sorting cells, it's the applications of this technology that raise those ethical questions. Stem cell research, for example. There are a lot of discussions about the ethics of using stem cells derived from embryos.
So it's not really the technology itself, but how it's used. that we need to be careful about. Are there any specific guidelines
Hmm.
or regulations to ensure that FACS is used ethically?
Yeah. There are. There are ethical guidelines and regulations for research involving human cells and tissues, and that includes those used in FACS experiments. These guidelines are meant to protect the privacy and safety of individuals and make sure that the research is done ethically.
That's good to know. It seems like with every big scientific advancement, there's always this balance between the potential benefits and the ethical concerns.
Yeah, that's right. It's important to have those open and honest conversations about these issues. As technology continues to develop,
Right.
public engagement and dialogue are essential for making sure that scientific progress aligns with our values and our ethical principles.
Well, today we've covered a lot, from the basics of FACS to its applications and the ethical considerations. I'm really amazed by this technology and the impact it's having. But before we go, I have one more question. What's the one thing you want our listeners to remember about FACS?
I'd say the most important thing is this. FACS is more than just a tool. It's a window into the hidden world of cells. It shows the complexity and diversity of life at a microscopic level.
That's a great way to put it. It's like we're zooming in on the building blocks of life itself, seeing all the intricate machinery that makes everything work, from our immune systems to how diseases develop, all that.
Right. And with each new discovery, we understand more about how cells function, how they talk to each other, and how they respond to their environment. And this knowledge, it's helping us develop more targeted therapies, personalized medicine, and ultimately just improving human health.
It's amazing to think how FACS is helping us unlock these secrets and pave the way for these new breakthroughs. So for our listeners out there, if they want to learn more about this technology, where can they go?
There's some great stuff out there online. In libraries, the International Society for Advancement of Cytometry, that's a good one. They have tons of information on FACS, educational materials, articles, events. You can also find good videos and articles on websites like Nature, Science, the National Institutes of Health, places like that.
Perfect. A great starting point for anyone who wants to dig deeper. Well, I think we've covered a lot today. The basics of FACS, the applications, the ethics.
Yeah, I think so too. It's been great sharing my thoughts with you and with the listeners.
It's been a pleasure. Thank you for joining us on this journey into the microscopic world.
Yeah.
And the technology that's changing how we understand life.
Thank you for having me.
And to all our listeners out there, thank you for tuning in to Podcast Learn and Fun. If you like our videos, please subscribe, click the like button, comment, and share. We'll see you next time.